Okay? It duplicates. All the DNA is exactly copied. Okay? So that is a true duplication. It's just like putting a piece of paper into a Xerox machine. There's an exact copy made. These two chromosomes, they all have the same genes on them, but they might not have the same alleles. Does anyone know what an allele is? What? Who's it? An allele is a different form of a gene. So let's say there's a gene that produces the pigment for eye color, an enzyme that produces eye color. It can exist in two different forms, brown and blue. Okay? It's the DNA that makes up that gene has a certain sequence, and it's slightly different in the two forms of the gene. So that gene makes, one allele makes one protein, and the other makes a slightly different protein. Okay? That's what alleles are. They're slightly different forms of the same gene. Okay? So, in most of us, we all have the same allele for the PKU gene that makes that enzyme. It's exactly the same. And therefore, we always make a good form of the enzyme that works. There are several mutations that can occur in that gene where you form an allele that makes a defective enzyme. Not all alleles are defective. Some can be just slightly different. But in that case, it's defective. It doesn't make an enzyme that works. So therefore, it's not very good. So, but now you have two chromosomes, each duplicated, and this one, they both have the same genes. This one might have different alleles than this one, but there are two copies. The first thing that happens in meiosis is that these two chromosomes come together and then at what's called the metaphase plate, and then they separate like this. And so you separate your maternal and your paternal alleles. Right? You separate them. Okay? Now with a one chromosome organism, it's pretty simple. Then these duplicated chromosomes fall apart in the next division of meiosis, and so you get 4 gametes, each of which is now called haploid because it only has one copy of each chromosome. You are what? What are you called? Diploid. Diploid because you have 2 copies. All right? So that's the basics of genetics. So in this case, let's say there's a different allele on the blue chromosome for a particular enzyme than on the red chromosome, right? What is the likelihood that the next generation is going to receive the blue allele versus the red allele? Can anyone tell me? What's the probability? This test says to me that half, let's say this is making sperm because it's in a male, half the sperm are going to be blue, carry the blue allele, half the sperm are going to be carrying the red allele, so that means it's 50%. 50%, 1/2.
Excuse me? Yes. On the cell phone? On the cell phone outside? Sorry.
No problem. Okay, so that's an important idea to keep in mind. The law of segregation. Alleles are separated in meiosis 1. That's this process. There are 2 divisions in meiosis to form gametes. The alleles are separated in meiosis 1. And the law of independent assortment says pairs align independently in meiosis 1. Okay, so what does that mean? We'll see now a little better if we consider an organism that has 2 different chromosomes, right? We have 23, remember? This one has 2. So, and normally in genetics, we show alleles by letters. So we say this person has the big G allele, this chromosome has the big G allele, and this one has the little g allele, okay, for the same gene. So this, and maybe one came from the father, one came from the mother, this from the father, this from the mother, and we say that this person has a genotype of big G, little g, right? Because that defines their genetic composition for that gene, right? And they are called what?
Heterogeneous?
Sort of. Hetero what? High heterozygotes. They're called heterozygotes. If you had both your chromosomes with little g, then you would be called a homozygote, right? So this person is heterozygous for this organism for the G gene and heterozygous for the W gene, right? See that? Okay, so now let's apply the laws of segregation, Mendel's laws, here. So in meiosis 1, we segregate alleles. Homologous chromosomes pair and then separate. Right? But wait a minute, guys. They pair independently of one another. So it's possible for both the blue sets of chromosomes that came from the father to be on one side, and the orange ones up from the mother on the other side. Or it's possible that they go like this, right? It's all a matter of chance. It's all a matter of chance. And this is going to happen 50% of the time, and this is going to happen 50% of the time. That's what's meant by independent assortment. So the different chromosomes assort independently. We're going to separate maternal and paternal alleles, but we're going to separate them for different chromosomes independently. That's really, really important to understand. And this is something that Mendel worked out. So the end result is that you'll get 1/4 gametes that look like this, 1/4 gametes that look like this, 1/4 gametes that look like this, and 1/4 gametes that look like this. And so that means that we have to consider those probabilities when we look at crosses. Now what we're going to do today is we're only going to consider what's called a one-gene cross. So we can ignore all the other chromosomes and what they're doing because we're only going to follow one set of chromosomes, right? We can ignore all the rest because we're not following them. If we did follow them, we would have to take it, and we wanted to account for all the different genes, we'd have to take them into account. Okay, so the tools of transmission genetics are things called Punnett squares, and then probability rules and branch diagrams, and then pedigrees. So let's look at PKU. The genotype is allele equals a version of a gene. If you have PKU, you are homozygous for a mutant allele of phenylalanine hydroxylase gene, sometimes called the PAH gene or the PKU gene. So you are homozygous for that mutant. You have to have count them, 2 bad alleles in order to suffer from the disease. Can anyone give me a reasonable reason why if you only have 1 bad one, you're okay?
Because there's a dominant allele that would surpass your recessive allele, which would be your PKU allele.
Okay, that's absolutely correct, totally correct. I will explain it slightly different. I will say that the PKU allele produces a defective enzyme. So that's on one of your chromosomes. Let's say you're a heterozygous, it's on one of your chromosomes. So you're making a bunch of garbage there, right? On the other chromosome, you have the normal allele, so it's making good enzyme. And it makes enough to get you through the night, right? So you survive. So we say that the normal allele is dominant over the mutant allele, which is absolutely what he said, right? We say it's dominant because if you have one good copy, you're okay. So that's called dominance. We say that the mutant allele is recessive. We say the normal allele is dominant, okay? That's not true for all mutant alleles. Some mutant alleles are dominant. And we say it's an autosomal recessive. So we say it's a recessive, this gene, because it's dominated by the normal allele. And we say it's autosomal because we mean it's not on the sex chromosome. That's all it means. It doesn't mean anything fancy. It just means it's not on the sex chromosome. So you have 22 autosomes, and you have one sex chromosome. The phenotype is mental retardation. So here we go. So here you see a cross between two individuals. One individual has big P, big P. That's the normal allele for this enzyme, right? And it is given a capital letter because it is dominant, okay? It's just standard notation. It doesn't mean anything special. And that person mates with someone who has PKU. If they have PKU, we know they have two recessive alleles, right? We know it with a certainty. And now they're going to get together and have children. And this is the cross. We're going to cross this times this, and all the children will be that. All right? They're heterozygous for this gene. We call them carriers. Why do we call them carriers?
Because they still have the gene, but it's just unexpressed.
That's right. And it can come out again in the next generation. All right? So that's easy. We don't have to do anything. All right. If big P, little p has the same phenotype as big P, then big P is dominant. That makes, that's what just sense. The genotype of all these individuals is big P, little p, right? Everyone agree with that? But the phenotype is expressed as P blank because it's ambiguous from looking at the phenotype